lecture. Yeah, thanks. I'm Dan Lewitt, in case you guys don't recognize me. I have the pleasure of today introducing our guest speaker, Michael Sughrue. And it's with distinct pleasure. I know Mike for a long time and have always been watching his progress and his trajectory. And it's really remarkable to see that he's really tackling important problems in neuroscience. Mike grew up in a small town in Oklahoma. He attended medical school in Columbia University in New York. There he graduated at the top of his class, completed neurosurgery training at UCSF, which is one of the most famed neurosurgery training places. I had the pleasure of serving with him as a tour of duty there. And subsequent to that, he actually went to trained with Charlie Teo at Prince Wales Hospital in Sydney, Australia, in endoscopic tumor resection techniques. He went to University of Oklahoma there, was a director of the Brain Tumor Center. He stayed there for about 6 years, performed remarkably 3,000 brain tumor operations. And at that time, was the busiest brain tumor surgeon in the US. He actually was recruited back to Sydney, Australia, and currently is a neurosurgeon at the same hospital that he was trained, which is an indication of how well they regarded him and his work. To me, I think his approach is relatively novel in terms of trying to dissect the brain connectomics of how things work together. I think this era, we have a lot of information, but certainly haven't had the chance to put things together and actually make a practical sense of it and application of it. And as a spine surgeon, and there's a lot of different decision makings in terms of how to address disease states, because everyone's a little bit different. And he actually attempts to make it in a scientific and deep process and learning method to arrive at a appropriate treatment for the patient. So certainly I await his talk with much interest to see what he has to teach us. So welcome, Dr. Sughrue.
Thanks, everyone. I'm going to get right into it. And this is a talk I've been giving as I've talked about connectomics for a while, and really thinking about why I was interested in this in the first place, but also talking about some of the things that it can do additionally. And what I'm going to try to convince everyone with is that the idea that utilizing machine learning, not as artificial intelligence to try to do our job for us, but to dive in and handle really large unstructured data sets and make them make sense, can help us make better decisions. Not necessarily answer all of the questions in the world, but make decisions. But given this is a talk that has an official title of translation, I want to talk a little bit about how to actually take a product into the real world. And Dan gave some of the background. As a disclosure, I founded a company. Normally that's a disclosure, but given that we're talking about translation, it's important to talk about what it is. And so, right now we have around just shy of 60 employees, but we're rapidly expanding after we closed the Series B to about 150 in 5 countries. We just closed a very large funding round. The company's valuation has gone up quite a bit. And we have an FDA cleared product and are taking another couple products through FDA and also multiple the regulatory. So again, we have some experience on how to take a really good idea that we're working on in the lab and figure out how do we make it a useful tool that can actually translate and work. So I'll talk a little bit about that too, because it's one thing to have a good idea, but no money, no mission. You have to think about how do you actually make this work. So I'm going to talk about neuroimaging and machine learning, but also how to take an idea and go out and make it happen in the real world. When to do that? It's not every good idea needs to be turned into a company. And again, when I started doing this research, that wasn't my plan. I'm actually still have that academic footprint. I still publish discovery papers, and not everything I do is necessarily directly related to the company. But also how to actually do that if it makes sense and why to do it. Especially why to do it if you actually really care about science and you're not just looking for a way to make some money. Okay, so the idea that I'm going to try to get across is connectomic neuroimaging, while people, we're still learning about it, is scientifically mature enough, and how to know that it's good enough to talk about translation. That's a tough topic, and no matter how you do it, when you first try to translate an idea, people will tell you it's too early. But that's, unfortunately, you have to make decisions. Um, that a lot of times that the barriers to why we don't get this on a daily basis are really logistical and translational. It's not because we don't understand the science, because we have a fair understanding of this at this point. And that ultimately, when you're translating, what you're really doing a lot of times is taking a fairly defined problem that we understand most of the boundaries of and best methods, and trying to figure out how to scale it, which in and of itself is a really difficult problem. It's just a different problem. And what I ultimately want to try to get everyone across is that once we get this across the finish line, it can have really profound impacts on decision making. So ultimately, I'm going to first start off by just showing you what we translated, in part just because it makes it easier. It gives everyone a frame of reference, because not everyone does this for a living. And so what it is ultimately is a cloud-based software. You see I'm running it in Google Chrome. And what the goal is that we wanted to make obtaining a connectome map, and I just typed my password in wrong, that could basically be done in less than an hour, around an hour, that was scalable, and it could be done at normal hospitals in the world. So it didn't have to have PhDs involved, didn't have to take 5 days, didn't have to take stringing together code. What we were aiming to do with it was take some of the anatomic research that I've been doing for many years and make it simple enough to where all anyone needed to do was say, I'm operating, for example, in the left insula. It goes off to the cloud, picks up the scan, the processed directly from the MRI. Nobody codes anything. And essentially what it would do is make an output that would put maps of large scale brain networks on an image guidance system. And so what we had to do is think about first, how do we use machine learning to find and make maps of brain networks? So for example, this is a network called the Salience Network. It wasn't known about when I was starting my residency by anybody. But how do we basically not only get it to make a map of an estimate of the Salience Network from the diffusion tractography, but also make it in a way, or for example, the language system, that it could export this to an image guidance system. And so essentially, there's a lot of parts in that, and it's more complicated than it sounds. But also, I want to point out that it's new to most neurosurgeons. So there's an ecosystem that you have to create around the topic in terms of medical education. The ecosystem you have to create around how do we even get this done when people aren't doing it? How do we change behavior and thinking on things? And that by itself could be an entire talk. But what the problem is ultimately trying to solve, and when you think about diffusion tensor imaging and why does it have neurosurgical approval, and what do people use it for? And it's a complex problem. And ultimately, if you were to talk to 99 or out of 100 neurosurgeons and say, which is more difficult, taking out a tumor at the base of the skull, like a really difficult meningioma, or an interaxial tumor in the right frontal lobe, almost all of them would say the skull-based tumor. And it is difficult because you have a tumor that's wrapped around nerves and arteries and things like this, and they're long and difficult operations. But those surgeons have one advantage: they can see all of their anatomy. All the cranial nerves are known, we can see it. So at least you have a fairly finite goal. It's just a difficult goal to achieve, but it's ultimately comes down to just operationalization. Whereas someone operating the right frontal lobe could be operating in a very redundant, safe part of the brain, but they could be operating in the thing that makes someone a civilized human being, and all of it looks like a white blob. And so it's an incredibly complex anatomy, and we don't know anything about it really at the end of the day. We don't know like anywhere what we need to know it. And more importantly, we don't get that information at the point of care. And so how do we take this completely amorphous anatomy and make it make sense and get that information in the best possible way when we don't understand everything we're seeing? And how do we gradually reduce that level of uncertainty, which is the problem we're ultimately tackling? And this is why I was interested in doing this in the first place. I didn't want to make patients into different human beings after I did an operation, at least as much as I could, given the fact I was treating oftentimes some terrible brain cancers. And ultimately what we're trying to do is we're trying to remove a tumor that is oftentimes and has brain function at least running around it or in its boundaries, or even sometimes through the middle of the tumor. And we know that gliomas and things like this infiltrate the brain, and there's often normal brain tissue in the tumor somewhere, but we have to often cut brain. I mean, if you look at this thing on the right, we're trying to get the patient from the left to the right with as few problems as possible. But if we don't know how they're wired up, or how the brain has changed plasticity, or how the tracks have moved, or whatever, then you're often going to stumble into problems and things that you don't understand. And ultimately, that's not very fulfilling if what you're trying to aim for is leaving people as human and normal as possible. And that's really the problem we're facing. But further, if you basically say that what your only goal is to not cause a neurological problem of any kind, including cognitive, the person on the left is going to get that no matter what. I mean, that's a horrible cancer. So we have to be brave and take care of these patients. But at the same time, the person on the right, it's hard to argue that taking out this person's speech entirely to get one more cc of tumor has a good risk benefit. And ultimately, this is a balance called the oncofunctional balance. We're trying to make this decision as finely grained as we can, given the situation. Can we save the function by, if we leave the tumor? Sometimes that is yes, sometimes that's maybe, sometimes it's no. We have chemotherapy options that are sometimes effective. And ultimately, we know that these tumors are often going to take the patient's life no matter what. And so we're trying to make a really difficult judgment call on a terrible situation. And we've kind of, it leads to this kind of sense of nihilism that we can't make a good decision. But of course, we have to treat these patients regardless of whether the neuroscience has matured enough to have absolute certainty, because these things keep happening and they're terrible. So a long time ago, I began trying to figure out how to do this better. So what we were taught was stay out of the eloquent cortex, and otherwise you're fine. But of course, even when I was a resident at UCSF, I noticed these patients' families would call me in the middle of the night with very distressed things. And ultimately it was that the personality had changed, or Johnny wasn't making good decisions. And while that's, at first I thought, this is 3 in the morning, I have people bleeding into the brain, no on call. The next thing I thought was, why does this happen? And how can we stop this? Because we know we can't get away with not treating these patients, but at the same time, how do we do a better job than this? And we were doing a good job, but we could have done better. So I started thinking about, how do we make cuts around brain networks? What are the anatomy of these things? And not a lot of people were talking about this at the time, in part because there was no way to get this imaging unless you were at certain institutions. We didn't know what a lot of these things did. The Human Connectome project and other major neuroimaging projects hadn't come as far as they had. And so if you didn't know what it really did, why did you care if you preserved it or not? And then ultimately it became a vicious cycle because we didn't learn from our mistakes. And so I just said, okay, these networks were probably important, and let's not destroy them if we can avoid it. So we came up with ideas of safe cuts. I wrote a book about this. This is what you're looking at. And so what I started thinking about is how to operationalize this. And I started going to meetings that were outside my area and learned about Connectomics, which is big data for brain connections. And while there's lots of ways you can do Connectomics, the one that really made sense is the thing that's likely to scale into the future are MRI-based techniques, not only because we know a lot about how to do them, but also because MRIs are everywhere. So this doesn't involve a bunch of hospitals buying new machines with increased capital costs in times when people are not necessarily interested in increasing their capital costs. So even if there's a better technique, it's one that we can probably make work because we're already getting MRIs for things all the time anyway. And one of the things that I encountered was people talking along the lines of, these are research techniques, and it really kind of surprised me, because when I looked into this, I found out there was about 550,000 papers on this. If you PubMed this has been studied out the wazoo, and we knew a lot about it. Everyone knows there's some limits, but there's limits to almost every test we do. And ultimately, there was this kind of fear to take this into the real world and say, well, how do we make these things useful? Because people thought, well, we don't know everything about them. And ultimately, we don't know everything about the human brain. That's a true statement. But as I pointed out, people still have the problem, whether or not we figured it all out yet. And so when I started thinking about this, and I was at the University of Oklahoma, and I didn't have a wonderful neuroimaging group like that exists at UCLA, I started thinking about how to get this done. And one of the challenges that I immediately came into was that the software solutions weren't very useful. It was very difficult for us to get this. Next thing I was learning how to code to do my research, and it just didn't work at the kind of time scale that I was working with. And ultimately, once I started doing this kind of stuff, even a little bit, I felt that there was at least some utility that we could figure out, but because we weren't getting it, we didn't know what to do with the information. And clinicians are really good once you give them a tool with figuring out, well, where does this fit into the milieu, or does it? And so I thought we needed to break this cycle. It's a pretty mature field, and try to figure out what could we learn from studying these techniques at the kind of scale on a regular basis in clinical practice. Now, ultimately, anybody who's thought about the Connectome knows that the challenge is that it's an extremely high dimensional complex problem. And so we've been getting pictures like this for a long time. And one of the things that I kind of felt was that people were paying a lot of attention on how to get the perfect corticospinal tract and fix artifacts, but they weren't necessarily trying to figure out what all these twigs did and what does this mean, and put this in an anatomically specific way that a neurosurgeon like me, who really needs to know cut here and don't cut here, would understand. So another problem is when we think about things like resting state fMRI, which has amazing clinical potential, given that you basically don't need to do a task, which makes it doable in the real world. It's very hard to get tasks done in a clinical scanner. Resting state's like ordering a flare. But of course, it's an unstructured data problem. And ultimately, if you think about the fact of taking the bold signal at lots of different areas and trying to make sense of who's talking to who and what does this mean, it's a big data problem. And while we know a lot about this, not much of it has actually been attempted to translate into real medicine. It isn't because people haven't thought of doing it, it's that people haven't just done it. Now, one of the best ways we know to cut down dimensionality in the problem of the brain is to parsilate it. We've known that since 1908 when Brodmann did it. This is the human connectome parsilation scheme. And people can argue about different ways to divide up the neocortex. But in short, we like this one because it actually fit with what I was seeing in surgery of about what the size of an area is, like how big is the speech arrest site. It's about the size of a parsilation. I also like the fact that it used familiar terms and consolidated other schemes, and it was very rigorous. So one of the things when this paper first came out in Nature in 2016, that immediately struck me was that we needed to use this to describe brain anatomy so that we could compare and talk about different parts of the brain. Because obviously what this can clearly show is that when there's areas that cross gyri, the brain doesn't care what we call a gyrus and a sulcus. That's why it doesn't have any consistent patterns. It actually just puts the function anywhere that it sees fit on the cortical surface. And we needed to adapt our thinking to address that. So I looked at this a little differently and just said, what we need to do is gross anatomy. And so I wrote a lot of stuff about this, mostly because I just wanted to know and to dig this down. And we did basically said what runs through different white matter pathways, who's connected to who most of the time. We didn't need absolute certainty, but basically who's connected to who. And what are these things kind of probably doing? And when I wrote that, I talked to a lot of my friends about this and I said, what do you think? And they said, this is an amazing amount of work, but how are you actually going to use this? No one's going to read all this. I thought that was fair enough because, like I said, I put months and months of work into this. And how do we actually use it? So in order to do this, what you have to do is you have to take an atlas and warp it onto the brain of the patient you're treating. And so you can see this is one way to do it, just a simple affine warp where you take, the atlas and rigidly shape it onto the brain, and it misses a few sulci. So there are other ways that people have that you unfold the brain and line it with polygons and use vertices to do this and other strategies. But the bottom line is I found that no matter what, if you put this patient into this, it didn't do a very good job of atlasing. And part of it is that this defies a lot of the expectations. Where are things? The tracts could have been destroyed or moved. If they had surgery, the brain is not topologically a sphere anymore. So how do you actually, unfold it to a sphere? And all the software failed or sometimes just crashed after hours and hours of processing. So this is really frustrating to me. So I kind of thought that we needed to basically be able to get something to work. And I've thought about a long time of how to do that. And when I talk to people about this, people always say, "That's interesting. I hope someone works on that for you." and so obviously we needed to have a solution. And so when I moved to Sydney, I met up with a guy who does machine learning, but he also had a PhD in neuroscience. So I showed him an idea I had how to do this, and he figured out how to actually make it a reality. So what we basically did was say, let's not make any assumptions of the shape of the brain. The thing that's topologically invariant are the tracks. So if you basically tie the parsilations to their connections, you could teach a machine learning model how to recognize those connections and know whether a voxel was part of, say, Broca's area or area 44, based on who it was connected to. And you could do it, this one, for example, would not be because it's connected to the wrong area. So that this would be a little bit more robust to this and be able to handle shape distortions. Because what it might do is look around here and say, well, that guy is not initially assigned to this, but it is. Now, there's some practical things of how to do this, of how to actually make this work. And I don't have time to go into all of the technology to do this, but what it does do is if there's a hole in the brain because there's a tumor with necrotic center, or there's a surgical cavity or whatever, there's no track, so it doesn't map there. And it allows you to handle some really abnormally shaped brains. And more importantly, it's really fast. So once you train the model, this can get done in less than 10, 15 minutes. So, and especially if you parallelize it in the cloud, you can do it quicker. But most importantly, it's shape irrelevant and can handle some pretty interesting stuff. And so this is the worst case I could find. So we wanted to see how it did against an edge case. And you can see that we've actually removed a good portion of the frontal lobe for cancer. The glioblastomas come back with a vengeance. This is not even a case we operated on, but because we were doing so much DTI at the time, one of the residents ordered it. So we told the patient, sorry, I don't think we can do anything for this. But then I had it, so I put it through this process. And so we looked at this and said, can it even handle it? Well, you notice that the insula is shifted, several centimeters ahead of the other insula on the side of the tumor, but the machine learning figured out that that's the caudate, that's the basal ganglia, and where everything was relative to that. Now, of course, with this, we don't know what ground truth is, but ultimately, it's a better estimate than having no estimate, and it also performs pretty well. So at least it gives us an idea of what might be going on, which is, again, used in the hands of a judicious clinician is better than having no information at all, which is what we have now, and gives us something that we can test. We can work on this, iterate it, find out what it's telling, what its limitations are, et cetera. And this is useful. Okay, so we have this technology, and the next question is, what do you do, right? So we could publish this, put this on GitHub, and then, give a couple talks and then hope that someone else ran with this in some direction. But some problems, once you get to a certain point, are not really very good academic problems. Academia is the light of the world, but it's not good at everything. So what academia is great at is unstructured problems. We don't know this. There's no framework for thinking about this. It may take years to sort this out. And academia has the kind of funding scheme and type of people and way of working that they're great at thinking about creative solutions to problems or handling problems that don't immediately have a use. There's no justification that you have to do financially other than this might benefit humanity. But it's not good at other things. And industry is good at things. And in many cases, the decline of medical research in industry has come at a cost. Because industry is pretty good at figuring out how to get things done, how to get things done in a timely fashion if they need to happen relatively quickly, and working on things that aren't necessarily sexy to grant committees, like putting a user interface on or making the software work without bugs or testing things in extensive detail to make sure that they actually work the way we think they work. And if you look at the quality systems that are required in industry, and safety and how it's really systematic and rational. I think that particularly academic medicine can learn a lot from the kind of stuff that industry does, because the systems actually are not as cumbersome, but they do their job very well. But more importantly, if we put this thing on GitHub, what if it doesn't work for somebody, or what if they can't figure out how to get to work? Well, that's kind of tough. If you can't figure it out, then it's survival of the fittest. But when you're selling a product, you figure out how to make it work for people. And when you're taking a novel idea into the real world, it just dawned on me that everything that I had heard of people proposing how Connectomics was going to change the world was not scalable, and that this was an industry problem. So even though I still publish academia, I still have an academic appointment, this is something we had to have a company really to actually make in real world. And one of it is that, if you look at the great software development, workflows, they're usually industry workflows. And part of it is you have to think about how software gets made. So someone comes up with a great algorithm. Okay, but that's the first part of how software gets made. The next thing you do is you give it to the engineers who don't care how the algorithm works, but their job is to make it run fast, make it work with other parts, make sure the software architecture is rational. And sometimes when you have 3 algorithms in a pipeline, they do something different than all of them do independently. That's emergence. And that is a very difficult thing to troubleshoot unless you have engineers who do that kind of thing, like figuring out that there's a rounding error that adds up between the 3 parts of the pipeline. It's very difficult. And then you link it to a platform, you make it run really fast, you get a user interface. And then if you're in the medical industry, you test it. I mean, you test it everywhere, but in the medical industry, the testing is off the charts, complicated. What I found in what academia is you get an algorithm, you just publish it, you maybe put a UI on it, just work on it a little bit, but most of the time not. And then you taught a course on it. But because that's how academics work, right? Everything downstream of that is not grantable. And so it doesn't really fit the workflow. And again, to show you that, again, all of this V, which is classic in industry of how you make a safe product, is not something you do in academia. Like, are we going to have detailed software architecture diagrams and prove every part of it works well and in combination, get testing, make sure it's usable? Just not really how academia works. Okay, so you get into a situation where you find something and you think, I have a good idea. I think this is something I want to do, and then think about starting a company. And having been around academia for a while, and a lot of great universities, been a lot of smart people, a good portion of these people kind of talk, off the cuff, like, oh yeah, I could have this company, or I might start this thing. And that's all great, but you have to just think about the reality. It's that starting a company is difficult. And I know we all are very talented people, but if you're time poor, you're not going to commit to it, or you think you just give the idea to a bunch of entrepreneurs and then go and give them some advice and it's going to work, it's a little bit unrealistic. So you have to start thinking about, how do you actually work through all of this to make a successful company? And of course, most of us would like a little bit of, money at the side of this. If someone benefits, it's our idea, we'd like to benefit from it too. But even if you don't care, you still want to also make sure that it works. We do these things because we want to help people. So how do you make sure that at the end of the day, that this ends up the way you want it to end up? And protecting and developing the early parts of this are actually the most critical ones, because this is where there's very few advice. There's lots of ways to screw this up, and there's very few people to tell you what to do. So I'll kind of give you some piece of advice. So if you have a good idea, the first thing is to stop talking about it until you've actually figured out inventorship and how to protect the IP and what that even means. You have to ask the magic question of all entrepreneurship, which is, who do you send the invoice to and why are they going to pay it? If it doesn't have some kind of way that at some point someone will pay an invoice, then it's not commercial. It's a scientific idea. That's you have to really think through that, and that's harder than you think. You think, oh, I know this will work, it'll be great, but it's a little more complicated than that. Even if you invented a brain machine interface that cures paralysis, you got to think how that's going to get paid for, because otherwise it isn't going to happen. If you don't have an answer to that, then investors aren't going to fund it. That's simple as that. It doesn't have to be tomorrow, but some point. Now, for drug pipelines, it's a little more obvious, but even then, still thinking through these things is really important. Who actually pays for this? Who actually orders it, and why will they do that? Assuming that your science is what you think it is. But also medical entrepreneurship goes a little further, because you can't send an invoice to anyone legally if you don't have FDA approval. So how do I actually convince people that the claim that I make is safe and that it probably is true? You don't have an answer to that, you don't have a medical device. So those two things are very difficult, and you spend a lot of time answering those questions, because you don't do this, you don't have a company, you don't have a product. And then you need to make sure you lawyer up and get smart people to give you good advice, because even if you don't care if you own 0% of this company, the tax liabilities for issuing yourself shares can be astronomical if you get the right amount of funding. And you really got to suit up. It's going to be some work. So let's go into all of these. If you start talking about things that you have a great invention of and you haven't figured out if there is IP in here, which there almost always is, and it can take years to untangle this. If you want to look at this, notice that Facebook, Mark Zuckerberg had a couple conversations with the Winklevoss twins and ended up spending $200 million getting them to go away. So it can be very complicated. And while everyone has good intentions, untangling a dirty patent can be years in court. So best to just make sure that you do this, because nothing stops somebody from hearing your idea and just filing a provisional patent in 2 days for $2,000. And now you have to spend years proving that you have discovered this and not them. And this is prior art. And again, they can just prevent you from developing the company. This is not about money ultimately. It's whether you can actually take this product and develop it. Now, IP is something that I didn't really understand very well until I met with really good IP lawyers who explained this to me. And so most people in academia get an inaccurate view of what is IP, protectable IP, what's not. And oftentimes we think it has to be this great invention that is so complicated that no one in the world's ever thought of this, and it was a brilliant idea. And that's usually not true. Sometimes it's just this is a technical solution to a technical problem. So this is a useful way to make a user interface that solves a problem that no one's thought of. That's a lot of IP. It's useful. It came out of your brain. And oftentimes the question is, who even owns it when it's thought up? So the first thing a lawyer taught me was that IP is created in the minds of humans and only is transferred to organizations by a contract. And so even if you think that, okay, well, I invented this, so everything's owned by the university. Well, maybe not, right? Depends on who else is an inventor, how this works, when it was invented, and what specifically is it invented, right? I mean, if you invent a better way to clean bowling balls, does it, does your job as a neurosurgeon necessarily count that? So it just depends. You got to think through these issues. And furthermore, who's an inventor, right? Because inventorship means a significant contribution to the idea that makes it that you couldn't imagine how you could have done this with this person. So when we invented this, the data scientist who helped me do this, we were both co-inventors. He didn't think of this before I told him basically a good idea how to work on, and I didn't know how to actually do that. So we both figured out how to make something happen in reality. And so you can't disentangle that, and you need to work through that and come up with a solution. But it doesn't necessarily mean that you outsourced to someone and paid them a contract and they brought you this great product and they didn't actually contribute any thought to it, or someone was at a lab meeting and said, yeah, that's a great idea, and then you're like, oh, let's get everyone on there, and there's some reasons for it. And it definitely doesn't mean that someone gave you data to do this, because if they didn't actually contribute to the idea, they're just a collaborator, then that doesn't mean they actually put any intellectual content into it. And while this kind of seems like this kind of petty line drawing, this can lead to some disasters. So I'll give an example. I met a guy who was starting a company in China, and he was a professor, and he was a very nice guy. And he basically split the invention 5 ways with his graduate students, even though it's his idea, they just did what he told them to do. And when everything turned into the company and they tried to commercialize this, it turned out that 4/5 of the company's equity was spent by these graduate students who didn't even join the company and weren't helping. It was dead equity. They didn't have any interest in this. It wasn't their idea, right? So now, not only did they getting a significant chunk of the money and all this other thing, but actually they diluted his control, because not only owns 20% of his own company before he's even accepted a dollar of investment. And again, this can lead to problems. These people could have equal stakes on the board. They may not even care about the company because there's nothing about this. And ultimately, this makes it so that almost the first day you take an investment, you're now a minority shareholder in your own company, and investors can just take it over and do something that you don't necessarily agree with. So ultimately, you really need to have these tough discussions, because while we're used to saying, well, let's give everyone credit, give credit where it's due, but also don't give credit where it's really not due. If you didn't actually add to the idea, you don't own the IP or have a claim to the IP. And these discussions are uncomfortable because people get their feelings hurt, but you have to have them, because at the end of the day, this can, even if you couldn't care less if you made $1 off this, if you don't control it, it doesn't work the way you think it's going to work. And ultimately, a lot of times it's important to know that you think that when you're talking about IP, that there's these big organizations and you don't have lawyers and you don't have rights and this sort of thing. And the ultimate thing is that ultimately what's in everyone's best interest is that someone does something with this that helps people and that actually makes money. So oftentimes you can talk to, have discussions and say, well, if I only have 5% of this thing, what is my incentive to actually kill myself to work on this? Because if nothing else, I don't even have control of this thing. And the best person to help develop the idea is the person who understands the idea, or at least contribute to it. So good IP lawyers can add a lot, but in addition to writing patents, it's strategy. It's thinking about where to file. How do you do this with the amount of money you have at the time you do it, especially if you're shoestringing your own company. Filing patents can add up to hundreds of thousands of dollars, and especially if you have a bunch of them. And how do you have more importantly, when you start this, and if this is successful, how do you make sure that Google or someone else doesn't copy what you do and push you out or sue you about it? So it's important to really think about strategy on how to protect your world so that you don't end up just, thanks for the idea, buddy, but we're going to just copy it. And, because ultimately that determines whether the thing can turn into reality. If you think you invented it, you're probably the best person to develop it, at least initially. When you ask for money, again, this is something that I didn't totally understand, which is, what do people who invest actually look at? Remember that these people are often really, really smart. Sometimes they have similar backgrounds to you, and especially if you're in med tech. But they have a different thought process because of what they're trying to accomplish. So what doesn't get funded is you're the smartest person who wrote a high impact paper in Nature. The idea is great. It's going to change everything. The data are crystal clear on this. You've got a great, smart group of people, and you're doing this in your lab all the time, or you give a plenary session talk. That's all nice. It helps your credibility, but investors will tell you up front that what they're not necessarily concerned about is that you're really smart, because what they can't do is fund indefinite science projects and lots of thought. What they actually have to have is that there's a coherent vision on how this could actually turn into some kind of return on investment at some point. And what's acceptable depends on the type of investor. So people who invest in Alzheimer's drugs have a long thought process, and people who invest in medical devices have a shorter one, et cetera. And people who invest in Silicon Valley, B2B startup, tech companies have a much shorter one. Now, you have to have a team that can realistically deliver this, but it isn't just smart scientists, but also people who know how to commercialize, product market fit. You have a good leadership team, et cetera. And so you really want to think about how do you get a team around that can deliver this and be coherent about this, but more importantly, that it's clear how the money is going to actually do that. How much money you need and why, and you have a clear plan on how you're going to get more money and when that needs to happen. And not just, well, we're going to apply for this round. Like, how are you going to do this exactly? What kind of milestones and progress are you going to make that makes someone interested in putting money in later? Because it's hard to do. So if you're not thinking about it, you're not going to build the company the right way. And ultimately, you have a clear idea how to get this through regulatory committees and how long that's going to take. And that this is some kind of thing that's acceptable for the type of investor you're talking about. Some people have long-term vision, other ones don't. But again, you're actually thinking about how to do this. There can be really early discovery science in the company, but it can't be an endless science project. That's just not why. And the reason is because there's a concept called burn rate. As soon as you start spending money and you're not making revenue, you're burning money. And so you either have to raise more capital, which dilutes everyone in the company, or figure out how to make a product that sells. So you think about burn rate like this, but this is actually what it is. You're building a plane while the plane is crashing to the ground. And so you have to think about really carefully, how do we keep the burn rate acceptable in a way that we can meet the milestones to either turn a profit or get the next round of funding. And to get that at where the company is appreciated enough where we're getting money for cheaper. And how do you value a startup company? Well, this is something that it's always hand waving. So ultimately, when you get to late rounds and IPOs and things, this is based on revenue forecasts and the usual stuff that a bunch of accountants figure out what the company's worth. But when you get to Series B, it's a little bit more vague, and we just closed the Series B, but it's clear evidence of progress, IP portfolios, that we've cleared regulatory burdens, things like this. And Series A, it's really vague, which is, do these guys seem like they know what they're talking about? Is this a good idea? Is it well thought out? Because Series A investment is high risk, but it also can be high return. But the more risk you have, the more expensive money is. And so you're ultimately trying to de-risk this proposal to these people so that they look at this and say, well, okay, it's not that high risk compared to what I normally invest in. And so we can talk about evaluation, and more valuation, the less money you have to, of the company you have to give up, and ultimately the less control. Again, at some point you want to make sure that your product actually hits the vision that you're looking for. And so you want to work with investors, you get what they're trying to get at, but you also don't want necessarily business decisions to drive the science. Now, ultimately, in the interest of time, you really want to sit down with this and go through how the board works, how things get voted on, how people get on the board, and more importantly, that you get to have a say in how things work, especially because you're doing this for a reason. And so really thinking through these things and making sure you have a smart lawyer who goes through and explains things. Again, you really have to make sure that ultimately this thing runs like you think it needs to run to an extent. And this is a negotiation, and you need to spend a lot of time talking about this. Okay. So as you can imagine, this is complicated. And so I think this is a good algorithm, which is, are you really going to spend, if you're going to do this, are you really going to dedicate this is most of the time, most of what you do most of the time? And so it's pretty simple. If you're really not going to be willing to spend a significant amount of time helping the people who develop the company learn how to get this science off the ground, because you're the person who understands that, and make them understand it and come up with help them do their job, then it's not going to work. And that's why a lot of times these side gigs don't necessarily go very far. But also, if you're going to do that, are you the CEO of this company? And the answer is, unless you really like to go out and raise money and do things other than science, it's good to get someone who actually is good at that. Now, let's talk about this and how, in terms of the product we made, how do we actually go about doing this? So we've got this IP, which is an algorithm for determining brain parcelations, but also there's other things in there about how it works and UI trick, how it handles data. And so ultimately what we're trying to do is solve a problem. You only can have a commercially viable product if it solves a problem, no matter how good it is, unless it's a miracle treatment. If you can't figure out how does it actually get paid for, it won't last forever. You can't stay commercially viable. So our idea was that we could get connectomic type neuroimaging on clinical scanners without PH expertise anywhere. Now, ultimately, we sat there and made a proof of concept in 3 months, and that de-risked our Series A round. We showed it to a lot of people and got feedback. I can't tell you how important that is. You learn a lot, and that we need to simplify it. And ultimately, we went back to saying, how do we take these montages and make it to where you could click a button, say, I'm in the lateral frontal lobe, and get this. And we worked through how to build workflows and engines and how to do that. And then the next question is, how do you, when you have this kind of thing, how do you know how to get through the FDA? So ultimately you think, I have to have this definitive, randomized, controlled Well, sometimes that is, depends on what it is. If it's a high risk device that's novel, sometimes that's what you need, right? But most of the time you don't. And so most of the time you need to take a claim that's circumscribed and you can prove it now. In our case, that we could do tractography and process it and atlas it, and it was reasonably accurate. And then find people to help refine the product and develop additional claims. And again, the nice thing about that second strategy is you're not coming up with something grandiose. You're saying this might be useful, and then having people who say, yeah, that seems useful, and having them figure out what the indications are, improving it. And this is something that you can do so that you're not constantly having to live in the world of raising money all the time. But it can go further too. And what we're working on now is how do you take the atlas, use it to take resting state fMRI, and find additional insight into things like functional connectivity, of how different parts of the brain are talking to each other. So a quick primer on functional connectivity. During a resting state scan, different parts of the brain are firing synchronously. Sometimes this is because they're part of a network. And by looking at the correlation, we can find things that are talking to each other more often than chance. And we can even, in our case, compare it to normative data of people who don't have disease. And what's interesting is while people have different activity during a resting state scan, sometimes there's areas that are too correlated or not correlated enough compared to normal people. And if you look at this for the whole brain, we know that people with mental illnesses of various types have interesting functional connectivity patterns. And based on this paper in Nature Medicine and many other papers, that this often links to clinical symptoms and defines disease-specific subtypes. Now, of course, whether this is the best model and things like this, or it's more complex than this, is an open topic. But that there's a functional connectivity problem, there's lots of evidence for this at this point. So ultimately, what we are aiming to do is take these kind of big data sets and explore the data such that we could leverage the existing information that we know about this to look at correlations and non-correlations, or even other forms of methodologies for looking at connectomics, which I don't have time to go into right now. And we built a giant hypercluster that basically figures out how do we model to learn where the connectome predicts things that we care about, like response to therapy. We built this to leverage the kind of machine learning workflows that are common in industry, like Google, Amazon type technologies. And the goal is to tackle the transdiagnostic hypothesis, which is, are there specific circuits that explain specific symptoms? And can we localize which parts of the brain in that patient are abnormal? Because of course, we could theoretically do something about it. So how do you even deliver something like this? Just discovering is great, but what are the doctors going to actually do? So we came up with a workflow. It's almost like asking an engine, where we would say, okay, I've got a patient who has schizophrenia and they have this symptom. Where in the brain does that usually happen? What are the options? And how do I look at my patient to see what that looks like? We built this big distributed hypercluster. It's able to process about 1 million scans at once in an hour. So it really just comes down to how much data you want to put into it. We can process more data than probably exists in the world. And we utilize some really interesting engineering workflows, what's called auto ML. And what that means is that once it gets the data, it models it using every conceivable machine learning approach with every single hyperparameter until it maximizes the area under the curve. So this allows us to, within 15 to 20 minutes, run 500 iterations on this model and find the best one. So this lets us fail fast and try lots of different strategies to figure out the way to model the data, with the eventual goal that what we would do is have a truly what's called an RDoC, which is a term that the NIMH brought to, of how do we figure out where in the brain these symptoms are, and thinking about the biology of these diseases in a mechanistic fashion as opposed to a DSM fashion. But also, more importantly, how do you make this make sense? And so thinking through these kind of things is where translation is, which is, what does this look like when a doctor gets this? It isn't just enough to find stuff and mine data, but how do you actually turn this into, where do you put these models, and what models should you make on what it's a lot of work. But of course, the goal eventually is that if you know where the problem is, you can do things about it. The simplest one is TMS, which is an FDA-approved treatment, but it also for depression, but it really is the data on this is that it can handle, it's very, very safe and can handle a lot of things. Of course, you can think of other things. I'm sure people on this thing, on this, call have heard all sorts of things about electrodes and BMI and things. And we all know that this kind of thing is coming in some form. Where do you put these things, or where do you aim these treatments? And that's a really important question, given that we know that these are extremely heterogeneous problems, and we need really complicated solutions. We can't pick a one-size-fits-all target. So just to show you some examples of what machine learning can do when you do it at this scale, this is some work on schizophrenia. We've mapped about 23 of the 30 symptoms. The ones we can't map are usually symptoms that happen in almost everyone. So if everyone has it, you can't make a machine learning model to predict when you don't have it. And so it's interesting when it tells us things that we expect, like the auditory and visual cortexes are abnormal in these patients who have hallucinations. But what do you make of this? And so the big question, of course, is if we make doctors smarter, the next question is, how do we make them smarter in a way that answers questions that we want to know the answer to? So I mentioned TMS, and it's a good treatment. We like it. It's not the only treatment, and we're not pushing TMS as a treatment because it's not our product, and we think that you can come up with other potential uses for what we're doing. But it's one we can use now, and so it's important to know that these data presentations that we see in papers have a real meaning in the real world. So if we look at this patient on the right who has a glioblastoma in the language system, we notice that there's a lack of correlation of areas between how the functional connectivity is disturbed, not surprisingly. Now, the interesting thing is that this is not just a nice academic exercise that we can see the biology of aphasia with resting state fMRI, which isn't that surprising, but that this is a place in the brain that we could potentially augment or do something to try to drive it in a direction. Now, what do we do is the question. That's a long question, but that once we can see it on an individual basis, even if it's noisy, we start to get an idea of how we could actually envision big data impacting medicine in the long term. TMS, you can do anything. So the biggest thing about we were aiming was, how do we make people smarter, at least have more information, and in a way that's actually usable and actionable. So part of it is actually taking the raw data, using tools to highlight what's not normal. So you can see on the right side, this is the red and blue squares on that matrix are the areas that are outliers of functional connectivity. You can see we figured out ways to handle multiple comparisons in a smarter way. And again, in the long term, the goal is that we can start taking patients like this. This is someone who's treated at our clinic, large stroke, 18 years old, a year and a half out, very minimal language improvement. And what we eventually figured out, if we looked at her connectome extensively, is that not only was her arcuate missing, but she had no communication between the supplementary motor area and the language system. And it was pretty significantly damaged. The stroke went almost to the midline. So we did TMS actually to the other SMA, which we never would have thought of. I would have immediately gone to Broca's area if I didn't have this kind of data. And not only did she improve significantly, but if you look on the right, that's the DTI of the same exact direction, the best we could replicate it 4 months later. So again, this was something that when Dan and I were residents was not something we were thinking about of how do we change the plasticity of the brain. But again, she is now back in attending college with some assistance. When this is something that probably wouldn't have happened for the stroke this big. And so we really need to think that these kind of things need to be sped up through the translation. In this case, it's better to have this kind of information and then figure out what it means than to not have it. And solving these kind of translational barriers have real world meaningful impacts. So thanks, everyone.
Thanks, Mike. Really wonderful talk and great advice in terms of how to actualize some of the ideas from academia to commercialization. Certainly very valuable advice for both myself and our trainees here. Couple of questions that, actually, anyone have any questions?